Hi, welcome to the Sybil Protein Search Page Screencast. In this screencast, we'll be covering how to use the Sybil Protein Search page. We'll use the Strep Pneumo website for this demo. Let's get started. First, we'll point our browser at the Strep Pneumo homepage. The web address is streppneumo-sybil.igs.umaryland.edu. This gets us to the current Strep Pneumo homepage. To get to the protein search page, we just click on the button labeled Protein Cluster Search. You must have JavaScript enabled for this page and for most all of Sybil. As you can see, the search page is divided into two sections. On the left is the protein search form, and on the right is the protein search results area. To start, let's focus on the protein search form on the left. The first field in this form is the search box. This box allows you to type in search terms and gene identifiers. Placing a comma between multiple terms will search for proteins that match any one of the terms. Next is the protein length filter. You may specify a minimum length, a maximum length, or both. These lengths are in amino acids. Next is the overlapping gene search. Checking the box labeled Find Overlapping Genes searches for genes that overlap other genes. You may also specify a minimum overlap length in the box below. This length is in nucleotide base pairs. The next section contains a list of features that have been found in the genome and on the protein sequences. Checking boxes here will return genes that contain or overlap the features specified. Selecting more than one will return genes that match any of the features selected. Next is the singletons filter. This section allows you to select genes that do not have membership in one of the computed gene clusters. In this case, you can specify that you would like genes that are not a member of an ortholog, a paralog, or either. Additionally, you may request that only genes that do not have blast hits be returned. Because clusters are derived using bidirectional best blast hit, a gene can be excluded from a cluster but still have a blast hit. Next is the organism filter. This section allows you to specify which organisms you'd like to search. Simply check the organisms from which you'd like to see results. Leave this section blank to search all organisms. The two buttons at the bottom will check or uncheck all of the organisms listed. The last element of this form is the search button at the very bottom. After you have filled in the items you'd like to search, simply press the button to get your results. Let's do a sample search. I'll enter protease in the search box. And I'll specify that I want genes with a transmembrane region. Then I'll click the search button, and my results should appear in the search results area. The results area can be expanded by clicking and dragging the border between the form and the table. The form can be hidden altogether by clicking the double left arrow in the form's header. It can be recovered by clicking the double right arrow. There are currently 6 columns in the protein search results area. They are number, which represents the initial ordering of the results, name, which is typically the locus name or some other gene identifier. This column's values are also links to the gene's report page, which will be covered in another screencast. Next is the organism name, sequence length, the length of the gene in amino acids, gene product name as per the annotation, and matches, highlighting the term that matched the search terms provided. Clicking on a column header will sort the results by that column. The arrow next to the term indicates the sort direction. Hovering over a column header reveals a down arrow on the right side of the column. Clicking on the arrow opens a drop-down which allows you to select ascending or descending sort. The Columns section of the drop-down provides a list of available columns and allows you to hide or show columns in the display. Unchecking a box hides a column. Checking a box shows a hidden column. Columns can be rearranged by clicking and dragging the column header left or right, and releasing when the blue indicator arrows appear in the desired location. At the bottom of the results panel are the pagination controls. Currently, the search results show 100 proteins per page. The number of results being shown and total number of results is shown at the bottom right. The number in the white box shows the current page. Entering a number into the box and pressing Enter will load that page of the results. The right and left arrow keys can be used to step through the available pages. The FASTA download button to the right of the pagination controls allows you to download a complete set of search results in a FASTA formatted text file.